Hi everyone, this is Chris. I'm a Master of Agriculture and Environment. Today I want to give you a presentation about my capstone project, Cytological Studies in 12 Ajuga plants in Australia, Limiaceae. My supervisor is Ruxin Yin, who is focusing on ornamental plant breeding in PBI. Okay, as shown in outline, we have 5 parts to talk today. Introduction, my project aims, and experimental method, result and discussion, and conclusion. Let's start from introduction. In today's society, nursery and gardening industry came into our vision because as the development of urbanism, more and more people are fond of gorgeous landscaping, and there is increasing awareness of climate issue. Thus, demand for nursery products growing very rapidly. And for such an industry, if they want to maintain its competitiveness, research and development for new products play a key role for the whole industry. And the research and development for new products could be like cultivating a new variety, thus finding plants with promising characteristics is the very beginning. Genus Ajuga is what I found. I chosen Ajuga as my material plant for the following reasons. First, it has unique ornamental traits, like its colorful and variegated leaves and colorful flowers from blue to violet. I even know a variety called Princess Nodia. It has tiny and pink leaves. Well, besides, Ajuga also have strong environment resilience. It could grow well from first sun to partly shadow, and it could survive on all soil types. Well, it is also deer and rabbit resistant. Well, for a long time, people are focusing on Ajuga's medical medicine, but nobody care about its ornamental functions. But as far as I know, it has already been used as ground covers. But all the varieties you can see on the market, they are derived from nature hybrid. Even for its 10 varieties which has US patents, they are nature hybrid. It is totally blank in Ajuga's artificial breeding field. However, the process of cultivating a new variety is very complex, including collection of germplasm, evaluation for promising characteristics, crossing, selection, and so on. So what my project has done is very basic. I observed the somatic capstone numbers of 12 varieties in Australia. There was no record of these varieties according to the capstone count database. So I hope that my project could provide new cytological information for genus Ajuga. Also, it may be an inspiration for future cytological investment like the karyotype analysis. Also, I hope that my project could provide background information for future breeding techniques, such as inter- and intraspecific hybridization and polypollinization, because these two techniques is very useful in breeding processes. My experimental was done in Plant Breeding Institute of University of Sydney, which located in Cameron, and 12 plant materials were selected for this experiment. The first is Ajuga australis, which is native to Australia, and 8 varieties from Ajuga reptans. Well, because Ajuga reptans is the type of species of this genus and has the most varieties among other species, and also have Princess Nodia, Chocolate, and one no-neighbor one. And here in this page, you can see some photos took from my experiment, the Hybrida Chocolate, the Ajuga tonally Princess Nodia, and Ajuga reptans Jungle Beauty. All the slides I used for observation was made by this chromosome dropping technique, and when I use this technique, I obey these three principles. Two to three slides of each species were made for observation, and if I cannot see any ideal chromosome or cells under the microscope, the whole process will be repeated to avoid some errors. Of course, some planned slides were made for several times, and I remember that the Black Scallop, the Jungle Beauty, and the Ajuga Australias, I made nearly 6 slides for them. And this is the schedule of the whole experiment. The study will start from September 2019. I made some preparations, and the major part is in November 2019. I made chromosome dropping and slide observation and recording the date. And until this year, I made the analysis, reflections, and some conclusions, also writing the research paper. And this picture shows the chromosomes in the Ajuga reptans purpurea, and the picture is not very obvious, but we can see that there are nearly 32 chromosome number under the microscope, and it was taken from my experiment. And this table shows the most important results from my experiment. As we can see, there are 8 materials have 32 somatic chromosome numbers in meristem cells from root tip, and there are 4 materials demonstrated an amazing number of chromosomes under the microscope. Because, and as we can see in the last slide, the picture could not be very obvious under the microscope, so it is really hard to count very clearly how many chromosomes in this species. So I only give a nearly figure here. So, and there was no B chromosome recorded among all materials. My reflections were around 4 respects. The first respect is the result I got. Most Ajuga reptans varieties have 32 chromosomes, which is in accordance with previous studies. But for those whose chromosome number over 32, I found a hypothesis said that the basic chromosome number of Ajuga is 8. If this hypothesis is accepted, we can see that Ajuga reptans is a tetraploid. The other 4 varieties, Ajuga australias, could be hexaploid with 48 chromosome numbers, and 3 varieties, the black scallop, Catherine giant, and jungle beauty, who are octoploid, which means that it has 64 chromosomes. The second respect related to the chromosome dropping techniques. The techniques I used refer to long study. Long used this technique on weight, so I think some modifications are required, especially focusing on the selection of pretreatment solution, the usage of the mini centrifuge, and its speed. The third respect is for further breeding study. I want to give suggestions that we can try some crossing between species with different chromosome numbers, because there was a study conducted on Sylvia, a close genus to Ajuga, and the study indicated that the differences in chromosome number alone is not a major barrier between the hybridization. And the last respect is about further study. I think further study may require molecular or genetic techniques to explain the mystery in Ajuga's morphological traits and genus diversity, because what I have done is very basic. Okay, here we come the last part, my conclusions. In my project, 8 varieties of Ajuga reptans and 2 new species, Ajuga australias, Ajuga ternary, were observed for somatic chromosome number for the first time. The somatic chromosome number of 8 materials is 32. The other 4 materials, Ajuga australias, Ajuga reptans, jungle beauty, black scallop, and Catherine giant, demonstrated far more than 32 chromosomes. There is a high possibility that they are hexaploid and octoploid, which has 48 and 64 chromosome numbers respectively. My project provides new cytological information for the genus Ajuga and background knowledge for further karyological or breeding studies, but I have to admit that what I have done is very basic. Further studies are needed. These are the references I used for this presentation. Okay, thanks for listening.